Hey guys, how you doing? So in this video we are going to look at the second half of the chapter, which looks at the rest of the cell cycle, particularly looking at cytokinesis, how that works, and then we're also going to take a look at what happens when the cell cycle goes wrong in the case of cancer, and then also have a look at stem cells. Okay, so let's get straight into it. Okay, so remember, in the last video, I sort of summarized the cell cycle and then looked specifically at mitosis and how mitosis occurs, and then we sort of got to the end of the mitosis phase, where now the cell has pretty much almost divided completely its nucleus and duplicated its nucleus, moved it to the other sides, and it's basically getting ready to divide the rest of the cell into the two daughter cells. It enters into the next stage, which is what we call cytokinesis. Okay, so cytokinesis is the division of the rest of the cell into your two daughter cells. Now, it's important to note that both mitosis and cytokinesis together make up the cell division process, but cytokinesis is not the division of the nucleus, it's division of the rest of the cell. Yeah, so it starts off being divide the nucleus in mitosis and then divide the rest of the cell in cytokinesis. Okay, so by this stage mitosis is complete and the cell has now two identical nuclei in it, but it's still one cell. The organelles, including the mitochondria and the chloroplasts, and also any other organelles in the cell, has moved towards each end of the nuclei. They're basically clustered around, pick their sides, and they're getting ready to divide. Okay, so in cytokinesis, you got to remember that each of those nuclei have the identical DNA to each other because the chromosomes have already duplicated and they've divided evenly. And both daughter cells are going to have identical DNA to the parent cell. Yeah, so each daughter cell, once it fully divides, will then enter back into interphase and will continue to grow and develop into its life cycle. Okay, and so you can sort of see there, after cytokinesis occurs, the daughter cells will then grow in size again until it reaches into the mitosis stage again, and it will then divide in half its volume, and then it will continue to grow and grow and grow until it repeats that process. So you can expect that around here the mitosis stage will begin. Okay, now in cytokinesis, particularly for animals, including humans, the process is fairly straightforward. The plasma membrane, which is what encompasses the whole cell, will actually pinch right down the middle and divide the cell. Yeah, and this process is called cleavage. Now, you got to remember the plasma membrane is not static, but it is a dynamic membrane. Yeah, remember that it's held together through the polar and non-polar forces of the phospholipids, and so when it pinches, it will then reattach and form this kind of cleavage furrow that's right here, that little groove right there. It'll cut its way through and form this cleavage and pinch into your two individual daughter cells. Yeah, and that's purely by the nature of the plasma membrane to be able to do that. Now in plants this happens as well, but it happens slightly differently because plants have that additional cell wall. So cytokinesis in plants needs to develop a new cell wall first in the middle before it starts to divide the cell. So what happens is you've got the same kind of end result of mitosis, but now you've got these kind of vesicles that are gathering in the center, and that vesicle then develops into what we call a cell plate, which is kind of your precursor to the cell wall. Once the plate solidifies and makes its way through the entire length of the plant cells, it will then form a new cell wall. The plasma membrane also pinches, and then you now have two daughter cells as a result. So you can sort of see there, cytokinesis is a pretty straightforward process. By now you've got your nuclei in each side of the new daughter cell, and half of the organelles will go into one half of the organelles will go into the other half, and the split for the most part is fairly even. Okay, so that's a pretty straightforward process in cytokinesis there. Now, For other cells, we're going to look a little bit at reproduction in particularly prokaryotes. You've got to remember prokaryotic cells are slightly different from eukaryotic cells, which is what animals are, in that they don't contain a nucleus or membrane-bound organelles. Okay, so they go through a different process of cell reproduction. So, things like bacteria and archaea go through a process of what we call binary fission. Binary fission is a kind of a modified method that is a bit more simplified, less complicated than that of eukaryotes, and because it's less complicated, it tends to be much faster. Okay, so instead of having chromosomes, chromatids, and chromatins like eukaryotic cells tend to have, prokaryotic cells like bacteria and archaea only have a single circular DNA strand, which is called a plasmid. Okay, now that plasmid goes through a very simple process. The DNA will replicate to form two plasmids. The cell and then begins to grow in size and elongate, and then it will then separate along the middle, and each daughter cell will form, and that entire process looks a little bit like that. It's a fairly straightforward process, and happens at a faster rate. The DNA will replicate itself, the cell will elongate, that kind of cleavage-like process will occur again, and then it will split, and the cell will form two new daughter cells, both of which have identical DNA to the parent cell. Remember, because prokaryotic cells don't have a nucleus, the nucleus doesn't dissolve, the spindle fibers don't form, and then the plasmid, the DNA, simply just moves to the poles as a result of that. Okay, and so the word binary fission there is used to describe this process, because binary meaning two, fission is to split. So binary fission is to split into two. Okay. Now, for eukaryotic cells, we use mitosis, but within the eukaryotic cells, the mitochondria and the chloroplast will undergo a similar process to this. And part of that comes back to, remember, in the endosymbiosis theory in chapter 2 that we looked at, where it was proposed that mitochondria and chloroplasts were once prokaryotic cells that were then absorbed and integrated into eukaryotic cells. So a very similar process will occur in the mitochondria of your cells and the chloroplasts within plant cells. Okay. Okay, so one of the key things to note is every time DNA is replicated, there's a chance that faults or mutations will arise. Now, a mutation is this kind of random occurrence or change in the DNA code. Now, for some mutations, it is harmless, but for some mutations, it can be potentially dangerous. And so it can interfere with the function of genes, including apoptosis, which is programmed cell death, also covered in chapter 2. And so what the cell then needs to do is that it needs to check and make sure that along the way, this particular process, this delicate process, the DNA that's involved in there is complete and damage-free as it gets transmitted to the daughter cells. Because once it gets transmitted to these daughter cells, if there is something faulty with the DNA, it can spread. And so the cell has to go through these particular checkpoints where it checks for faults to make sure that the process is smooth. Okay? And so there are a few molecules that are involved in this process. These molecules are called cyclines or cycline-dependent kinases. Yeah? Now, both of these molecules are different families of molecules which perform similar functions in what we call the gatekeeper molecules. Yeah. So what they do is they regulate the cell life cycle. They will either promote and sort of push the cell through its life cycle, or they will stop the cell from progressing through its life cycle, depending on whether they find any mistakes in the DNA code. Okay, and so these gatekeeper molecules will check the cell at particular stages in its life cycle. Okay, so 3 in particular. At the end of the G1 phase, there is a checkpoint. At the end of the G2 phase, there is also a checkpoint. And right in the middle of the mitosis M phase, there is also a checkpoint. Okay. Here's an overview. Now that red dash tells you where the checkpoint occurs. The first one occurs right at the end of the G1 phase, after the cell has grown and prepares to replicate its DNA, these molecules will then kick into action and they will stop the cell progression and check for cell size. It checks that all the nutrients is there for the DNA to replicate. It also checks that the DNA isn't damaged before it's replicated. Okay. Now, round about this stage as well, if the cell is entering into quiescence, remember that is that resting state or that normal cell function, then it will exit around about this checkpoint as well into the G0 quiescence phase. But that's the first checkpoint. The second checkpoint happens right before mitosis. So the DNA has replicated at this stage from the S phase, and the cell now doubles in size, and right before it's ready to divide that nucleus to split that DNA into its two daughter cells, it will do one last check to make sure that it's big enough for one, and also that the DNA that's replicated is accurate and there's nothing faulty that has arisen, because that replication process isn't always perfect, and mistakes can occur during that phase. So once again, these particular molecules, gatekeeper molecules, will kick in and just check the life cycle, pausing it or promoting it at that stage. The third one is right in the middle of the M phase, where it will check that the chromosomes have attached properly, so that once the cell splits, the chromosomes are in the right place that they are meant to be in. And as we look through this chapter, and even the next chapter, you'll understand why that is a particularly important phase. Okay, so let's take an overview of each of these checkpoints. The G1 checkpoint happens in that G1 phase. It checks the DNA for damage, so it needs to make sure that the DNA is damage-free before it replicates that DNA, because once it replicates the DNA, that damage becomes replicated and passed on. Okay, the cell also needs to have enough nutrients so that in order for it to divide, the nuclear membrane and all those things need to have sufficient nutrients, the centrioles, the spindle fibers, and so forth. The cell then needs to grow in size, and that those patterns are normal. At the G2 checkpoint, after the cell has replicated its DNA, the DNA is then screened for structural damage. In other words, the actual individual parts of the DNA is complete and perfect, and there are no mutations or faults occurring. If there is something wrong, it will pause at this stage before it enters into mitosis, so that the repairs can be made to the DNA. It also checks that the cell is large enough so that it can divide. Okay. The third one is right in the middle of the mitosis checkpoint, and it happens right at the end of metaphase and anaphase. Now remember, at metaphase, the chromosomes have lined up along the equator. Yeah. During this stage, the cell will hit pause temporarily, and it will check that the chromosomes have lined up well and that there are no defects in the attachment process. Now, the reason why this is so important is because once the chromosomes are split, they will move into their respective sides, and that sort of is the kind of the point of no return for that cell. And so it needs to make sure that the chromosomes are lined up perfectly so that no chromosomes are moving into the wrong cells. Otherwise, what's going to happen is your daughter cells are going to have an uneven number of chromosomes, or it's going to have an uneven distribution of the DNA that's in there. Okay, so it needs to make sure and check that it's lined up perfectly and the chromatids are separated. Okay. So that's one of the ways that the cell can check for something to go wrong. Now, there's another way the cells can check to make sure that nothing goes wrong, and that's what we call genetic control. Okay, so these types of genes play a major role in controlling the cell cycle. Okay, particularly in kind of the occurrence of things like cancer. It's related to a lot of these genes. Yeah. Now, the first class of genes we have are what we call proto-oncogenes. Okay. Now, proto-oncogenes are genes that will stimulate cell division. Yeah. What that means is they want the cell to divide, so they push the cell forward through that division process. And what it does is it stops the cell from differentiating or regulating its own programmed cell death, and it continues and it promotes growth. Yeah, and that's what it wants to do. It accelerates the cells to grow and divide. So one of the key questions you then need to think about is, well, what happens if something goes wrong with these genes? If these genes don't work, what that means is the cell is not going to be stimulated to grow and divide, and it's not reproducing through mitosis and not performing that growth, development, repair part that is really important in the overall well-being of the organism. Okay, so those are one set of genes that play a really important role in regulating our life cycle and things like that. Okay, now the second one are what we call tumor suppressing genes. Now the tumor is this uncontrolled growth, so a tumor suppressing gene is a gene that suppresses growth within the organism's cells. They inhibit cell division. So you can see there, the proto-oncogenes and the tumor suppressing genes do kind of like the opposite thing. The proto-oncogenes will stimulate growth. The tumor suppressing genes will inhibit growth. They slow down cell division to repair DNA mistakes, or they will stop it altogether, depending on the situation. Okay, and so you can sort of see that during things like cancer, these tumor suppressing genes, which are preventing cell division, are no longer functional during cancer, because cancer is uncontrolled cell growth. It is when the cells are growing too much. Okay. Now let me mention one particular type of tumor suppressing gene called, which is often regarded as the guardian of the genome, as an example to talk about how your cells actually regulate and guard against things like cancer. Okay, so there is a particular gene called the p53 gene. Now this p53 gene is considered as the guardian of the genome because it plays a really important role in producing a type of protein called the tumor protein p53. Yeah, this is a tumor suppressing protein. The protein performs a vital function in the life of a cell in your body, and what it does is it detects damage in the DNA, and what it does, it then triggers a response. It's kind of like the commander-in-chief in your cell that regulates the DNA and makes sure that it is replicating properly. Okay, so here is a diagram of what happens. So normally, if your cell is doing really well, it's replicating, the DNA process is perfect, it replicates, then at those checkpoints, this p53 protein isn't activated, and so it progresses through your cycle, and your cell will go through mitosis, it will divide, and it will form your daughter cells as per normal. However, let's say at one of those checkpoints, there is a damage that is detected in the DNA replication. So there is some sort of DNA damage that occurs. Okay, what will happen is between this G1 phase, or within the G1 and S phase, that checkpoint, the p53 gene will activate. Yeah, what it does is then it pauses the cell cycles. It stops the cell from entering into the S phase, and it will prepare to fix that particular damage. Yeah, so two things have happened. One of two things will happen. So the first is that the DNA is going to be repaired, so the enzymes will be activated to initiate DNA repair. So what happens is it will stimulate and regulate more enzymes to move into that area and try and fix that problem. If it is a mistake in one of those nucleotides, an A, a T, or whatever, it's going to try and fix that. Okay, if it fixes it and it is successful, then the cell cycle will then resume, because the damage has been repaired, and it will continue to cycle through the S phase, the G phase, enter into mitosis, and so forth and so forth. Okay. However, what the p53 gene will also be able to do is that if the particular damage is too severe and cannot be repaired, what's going to happen is the p53 protein is going to induce apoptosis. Yeah, it's going to force the cell to self-destruct. That is, the lysosomes are going to rupture, and the cell is basically going to release enzymes on itself, and it will be destroyed. Yeah, and the cell will die. Now, this is really important, because if the cell is damaged, it needs to be terminated because it's going to divide, and that damage to the DNA is going to be passed on to the daughter cells. And remember, each parent cell will produce 2 daughter cells, which will then duplicate again into 4, 8, and so forth. And so that DNA problem needs to be identified and repaired. Otherwise, the cell needs to be terminated so that that particular damage doesn't spread. Yeah. So it's really important. So the p53 gene plays a really important role in controlling the cell cycle. Yeah. So you can imagine that in the event of cancer, that particularly if there is a damage to the p53 gene, that there is a kind of an association of damage to the p50. If something goes wrong with this p53 gene, then you're going to get massive problems around the development of cancerous cells and tumors in the organism. Okay. Okay, let's take a look at cancer and talk a little bit about how cancer works. Now, this is actually taken from the Cancer Council website, and it sort of talks a bit of an overview of what cancer is. In very simple terms, cancer is really just uncontrolled cell division. It is when your cell divides uncontrollably. It's not stopping in its division, and it's becoming too big. Okay, and so your normal cells will divide within a certain kind of time frame. When the cell dies or damaged, or it reaches the end of its lifetime, it will duplicate, and then it goes through cell division. However, if there is some sort of mutation in these genes, it's going to cause the cells to divide too rapidly, which then forms what we call a tumor. Yeah, the tumor is that little cluster of cells that are dividing uncontrollably. Okay, and so the abnormal cells will begin to multiply, and that tumor is that cluster that's there. Okay, and then what can happen is 2 things can occur. That tumor can either be benign, which means it's what we call localized, sitting in the same place, anchored to a particular tissue in the body, and it doesn't spread. Okay, that's a typo. It's meant to say tumor cells doesn't spread, right? Oh, yeah, I suppose it could also say don't spread. You can tell my English is not that awesome. Okay, now the other thing that could happen is, if it's not benign, it could be what we call malignant. Now, malignant tumors are what we have to watch out for, because they're the ones that are considered as what we call cancerous, because these tumor cells are not anchored to a particular tissue, and they can spread to other parts of the body. So they can enter into the bloodstream or the lymphatic system, and it can travel through the lymph or the blood into another area, and when it gets into another area, it is invasive. In other words, it causes those particular cells to then also become tumorous, and you get multiple tumors forming around different parts of the body. Okay, and so this is that kind of cancer stage that individuals sort of enter into when a malignant tumor forms. Okay, and so that spread will happen through the lymph vessels or through the blood vessels. Oops, so that's the same cells, same slides. And you can sort of see there, what tends to happen is that tumor growth forms, and then it breaks, a section of it or a portion of it breaks off, enters into a transport vessel like a lymph node or a blood vessel, and then it moves to a different location where it can then infect other cells, altering their DNA and causing them to also become cancerous as well. Okay, so that's one of the key things that you have to sort of be careful and screen and check whether or not a tumor is benign or malignant. Okay, and different cancers are named depending on the location of the cancer, but in the end, all cancer is really just uncontrolled cell division.